Okay, it's good. Great. Thank you.
So thank you so much. I have been I've been at the Sanger Institute for over 3 years now. And as Imran has said, We've worked on across various pathogens, so strep pneumo and strep agalactis, specifically on the training aspect. I did also a bit of research, and now I've moved on to the cholera genomics work and working to put together a community of practice, and also that involves capacity building as well, and using genomics to inform cholera control. And I thought to myself, so when I got this invite, I thought, what should I present, especially having shift various fields. And we did talk about presenting on cholera, but I felt it's still on the early stages. And as I continue to develop ideas and work with our collaborators, maybe I can be able to share what I've been able to do at a different forum. But for this aspect, I thought I could also share another aspect of what I've done, which is on hepatitis B virus drug resistance. So looking at its biological foundation, epidemiology and impact on public health. And this work, there is a lot of sequencing and genomic data analysis that went into place. And as you know, we are thinking on pathogens more broadly, there might be some overlap in terms of thinking how we analyze and handle various data sets. Um, so I have nothing to declare, and so I'll move on to my presentation, and I'll begin by giving an overview of its burden. So chronic hepatitis B virus infection, it remains a major global health challenge despite the availability of effective treatment and vaccines. And really, it is with these interventions in place that the global strategy aims to eliminate viral hepatitis by the year 2030 by upscaling vaccination, diagnosis, and treatment efforts. However, there are barriers such as limited access to diagnosis and treatment in low and middle income countries, and also the lack of robust epidemiological and genomic data does hinder our progress. And so a key question here is, where does hepatitis B drug resistance really fit within these broader public health challenges? And just reflecting on the pneumococcus, these are also similar challenges where we have an effective vaccine for strep pneumo, but there are challenges with, around, infections still occurring even with these interventions in place. And so this other slide, I will take a step back really to give an overview of what hepatitis B virus is, and then we can start understanding drug resistance, and I'll take you through its biology. So hepatitis B is a DNA virus that infects the liver, causing acute or chronic liver disease. Its genome is organized into a partially double-stranded DNA of about 3,200 base pairs, containing 4 overlapping reading frames that encode for viral proteins. And so you can see this blue, green, brown, and yellow lines just show how the genome has all these overlapping reading frames. And so the blue one will encode for the polymerase enzyme, the green one, the surface proteins, the yellow, the X proteins, and these are the other core proteins for the virus. And so when hepatitis B infects the liver cell, it binds onto this NTCP receptor, a receptor releasing its genome into the cytoplasm, where then it's transported into the nucleus, where this now partially double-stranded DNA. Sorry, can you see my cursor, if I may ask?
Yes, Dr. Jolynne, we can see it.
Okay, good. Okay, so yeah, so when the virus infects the liver, it attaches onto this NTCP receptor, and then this it moves on to the nucleus, where this partially double-stranded DNA is converted into this covalently closed circular structure, cccDNA, which is the root cause for Hep B chronicity. So this is cccDNA also serves as a template for transcribing both mRNA, these blue things here, and the pregenomic RNA, this green shown here. So moving on to the cytoplasm, these mRNA are translated into viral proteins, including the polymerase enzyme, and then the pregenomic RNA is reverse transcribed by this polymerase enzyme into this partially double-stranded DNA, which can be utilized in two ways. It can either go back into the nucleus to replenish this cccDNA pool, maintaining this aspect of the virus, or it can be released to infect other new liver cells. And so the persistence of this cccDNA is the root cause of Hep B chronicity. And there's no drug that targets this cccDNA, and this explains why hepatitis B virus infection has no cure. But to prevent or manage the disease, current treatments do target this viral polymerase enzyme, therefore inhibiting viral replication. But then, so the key challenge here is that Hep B is not curative, and this reverse transcriptase, the treatment targets this reverse transcriptase enzyme inhibiting viral replication. And so drug resistance can be one reason why treatment may fail to suppress a viral load, where there is resistance within the viral polymerase enzyme, and therefore when the drugs are administered, they are not able to bind onto this enzyme, and therefore replication can continue to occur. So that is one way. But then there are several aspects, including inconsistent access to medication and socioeconomic or psychological barriers that can actually hinder treatment adherence. And these challenges taken all together can make individuals who struggle with regular treatment more vulnerable to developing drug resistance. So examining the current antiviral treatment options, we say these drugs do target the viral reverse transcriptase enzyme inhibiting replication. And so this drug, this here shows a list of drugs that have been used for hepatitis B virus infection. Lamivudine has been widely used in the past, but it is really currently limited because of resistance. Today, entecavir and tenofovir are the current recommended treatment options for chronic hepatitis B virus infection. Tenofovir, unlike entecavir, tenofovir is relatively inexpensive and widely available in Africa. It is also effective against HIV and can be safely used during pregnancy to prevent mother-to-child transmission of hepatitis B. However, while tenofovir is accessible as part of the antiretroviral combination therapy, see, a lot of these drugs used for hepatitis B infection control actually were initially developed for HIV and are still used for HIV treatment. And so these drugs are they're available as combination therapy, and in some cases, it's really unavailable as a monotherapy in several regions. So you get that people who are HIV Hep B co-infected have a better chance of accessing treatment, but those who are hepatitis B mono-infected necessarily don't have, because some of these drugs are, tenofovir as a drug in itself is not readily available in some setting. But then given this consideration, so a key question here would be, how does resistance impact the effectiveness of these current treatment options for hepatitis B? And so my PhD work focused on advancing hepatitis B elimination, specifically addressing the issue of drug resistance. And I began by reviewing existing epidemiological data through systematic literature review, which examined the frequency, occurrence, and distribution of resistant mutations in Africa. So this review included 40 studies And this map here illustrates the countries where these studies were conducted, and the boxes represents the individual mutations reported across them. Majority of this of the study populations were HIV Hep B co-infected individuals, and lamivudine was the most commonly used treatment. One key finding from this analysis is that indeed there is limited data from Africa, with the entire region, such as North Africa and also this southwest part of Africa, being completely unrepresented. Additionally, the data that I gathered was highly heterogeneous, which means that there are various different populations included. The sequencing approach that was applied across these studies were different. Some people specifically for sequencing one section of the virus, where others are sequencing the whole genome. So there was that kind of heterogeneity across these studies. From the mutations I identified, so the key thing here we are I'm looking for mutations located within this blue region here, this polymerase enzyme, because the drugs they target the polymerase enzyme, therefore inhibiting. But if there are mutations across this this section of the genome, then that could help us understand resistance. So I observed so I identified studies that reported resistance to tenofovir, as well as entecavir, this ETV here, and there's also mutations associated with lamivudine. There are also vaccine escape mutations associated with the surface protein. But really, Hep B resistance to tenofovir is a controversial topic. While some studies actually report no resistance even after several years of using this treatment, there is emerging evidence suggesting otherwise, as shown in some of these studies. And so my next step was to investigate whether Hep B resistance to tenofovir truly exists, and if it's so, to understand the underlying mechanism behind its emergence. So I conducted a second systematic literature review to assimilate current evidence on the clinical and virological breakthrough of hepatitis B virus during treatment with tenofovir. So I included 15 studies that used various methods to define resistance. There are some that sequenced the virus isolated from individuals not responding to treatment, while others employed in vitro assays to measure the effect of tenofovir on viral replication in cell line. And there are some studies which combined these two methods to assess for resistance. So they looked for resistance. They recruited individuals who are not responding to treatment, and they also tested these mutations identified from viruses isolated from these individuals to test whether the virus replicates in vitro assays in the lab when exposed to treatment. And so the Hep B resistance to tenofovir emerges in the context of multiple mutations, with the most commonly mutations reported at position 180, 181, 204, and 236 of the polymerase enzyme. So my next step in this analysis was to explore the structural impact of this mutation. So looking at when you take the Hep B polymerase enzyme, the structural the crystal structure of this enzyme. So this here is actually crystal structure for HIV polymerase enzyme, and the reason being is that there is actually a lot of homology between both HIV and Hep B polymerase enzyme, and that's the reason why drugs used for HIV treatment can be effectively used for Hep B treatment. And so I mapped these mutations onto this solved crystal structure to infer their mechanistic and structural consequences. And so this map here revealed that most of these mutations associated with resistance are situated spatially adjacent to this gray structure here, which is the critical core of the enzyme. So if you can see this position 204 here, it's quite close to this gray region here, which means that any changes within or around this region can actually change the structure of the enzyme, interfering with its fitness, but the virus compensates for this by acquiring mutations in other regions to restore its fitness. So Hep B resistance to tenofovir does indeed exist, but it exists in the context of multiple mutations located within the active site of the enzyme. And so the next question is, how does this actually matter? And should our understanding of drug resistance influence broader treatment strategies? And so to provide a global overview, I analyzed 6,000 Hep B sequences downloaded from a public database to describe the global prevalence and distributions of these mutations. And so this figure here highlights this mutational position 204, which is well known to cause resistance to lamivudine and also associated with resistance to other antiviral drugs. You can see that in some population, this mutation has a prevalence of up to 5%, which could potentially interfere with therapy at a population level. However, when it comes to mutation, especially when it comes to when you combine these mutations, the frequency is much lower and they're less likely to affect treatment at a population level. As shown in the previous slide, Hep B resistance to tenofovir does require a combination of mutation, but there are still gaps in understanding its prevalence. So having looked at the global prevalence of individual mutation, the next step was to assess the global prevalence of clinically significant resistance to tenofovir and entecavir, the current treatment used for Hep B. We conducted another systematic literature review and meta-analysis, including 62 studies with approximately 12,000 participants. These studies reported resistance based on viral sequence data. And so for entecavir, resistance does increase. So if you look at entecavir, this section here, it does increase over time, reaching an average of 9% in those who are treatment naive and up to 20% in those who have been exposed to previous treatment within 5 years of treatment. And then when it comes to tenofovir, the risk is actually much lower. You can see it's actually at 0% at all times, whether people have been exposed to treatment before or not. So from this analysis, unlike entecavir, tenofovir does demonstrate a robust resistance profile, although data gaps may underestimate the true risk of resistance in real-world settings. So look, we've previously looked at the population level, but when you look at this mutation at an individual level, they really can have significant implications when you look at an individual. So we did conduct a cross-sectional analysis of a South African cohort of individuals living with chronic Hep B infection. And there was this one case who was both HIV and Hep B co-infected, who had been on treatment for over 60 months. So this figure here shows the distribution of their HIV viral load, which is shown in this blue color, their Hep B viral load, which is shown in this maroon, and then this yellow and green shows their liver enzyme, which actually suggests ongoing inflammation. So you can see with treatment, their HIV viral load does decrease over time, but when you look at their Hep B viral load, it is still elevated. You know, it is still high despite this individual being on treatment. And also, when you look at this elevated liver enzyme, it actually shows that there is ongoing inflammation. And so, based on this observation, we did suspect that the reason why this individual is not suppressing their viral load, hepatitis B viral load, despite being on treatment, is that it is because of drug resistance. And so we sequenced the virus and we were able to identify 9 mutations associated with resistance. And so this case here highlights a clinical phenotype of an individual not responding to treatment and a viral sequence phenotype showing mutations linked to resistance. And so the next step here is to test these mutations in an in vitro assay in the lab to assess the impact of tenofovir on viruses carrying these mutations. And this work is currently ongoing at the Crick Institute in London. So from this analysis, hepatitis B virus drug resistance may not pose a widespread population level challenge, but it remains relevant at an individual level, especially for those with treatment failures. However, data gaps such as limited data from low resource settings, key population, incomplete clinical metadata, inconsistency in laboratory data, and a lack of standardized resistant interpretation really does impede comprehensive insights of hepatitis B drug resistance. And so moving forward, clinical and public health solutions should include integrated access, accessible, cost-effective, and scalable hepatitis B drug resistance testing, ensuring that testing is widely available and affordable, enabling timely detection of resistance and appropriate treatment adjustments. And then the second thing is implementing robust data collection methods, establishing standardized, reliable systems for collecting and analyzing data for Hep B prevalence, looking at the resistant patterns as well as treatment outcome. And we also need to expand testing and data sharing network, which really is a common theme across all pathogens, really strengthening collaboration between healthcare facilities, research institutions, and public health organizations to share data and resources, enhancing surveillance and response efforts. And finally, optimizing treatment strategies by developing and implementing evidence-based individualized treatment plan that consider resistance patterns, co-infection, and other factors to improve patient outcome and also reduce transmission. This work has been done with a lot of guidance from my supervisor, my PhD supervisor, Professor Philippa Matthews, who's based now at the Crick Institute. There are a lot of funders involved in this work, and also a huge team of individuals who have contributed in one way or another, and also collaboration across multiple institutes across the world. So that's it for now. Thank you so much for listening, and I'll be very happy to take any questions. Thank you.
Thank you, Jolene, for an excellent presentation. Very enjoyable, very thorough. And we will now open for questions from comments from the audience. Any questions and or clarifications or comments from the audience?
Hello, one of thanks for the very nice presentation. As your presentation suggests, focused work was done on particularly on the genotype C, right? And that actually in context of resistance with the tenfold. So, is there any data that actually that has been done? It's the resistance of tenofovir in other serotypes like type 1, 1A, and other?
Julie, were you able to hear the question?
Not clearly. So if someone can.
My question is that the work that you just presented actually spans and actually evaluated the resistance in context to particularly the genotype C of HBV. Am I right?
Yeah.
So is there any data that or similar study that has been conducted on genotype 1 or other genotypes of the HBV that shows how the effectiveness of the efficacy of the tenofovir in this the other genotypes?
Yes. So thanks a lot. So Hep B genotypes are they are distributed across different regions. So in Africa, you get like these genotype A, E, all that, and in some settings, for example, in Asia, you get that there's genotype B. But what we did, we included all so for the global prevalence, where we looked at publicly available data sets. So that included data, and it represents all genotypes that were present, and we're able to see how resistant the prevalence of resistance varies across all these genotypes. And you get that, so for example, if you look at one specific mutation, and let me just share my screen again and show you. So when you look at this figure here, and it shows the prevalence of individual mutations across each genotype. And when you focus specifically on this mutation that has been shown to cause resistance across various treatment, it has the prevalence varies across all genotypes. But the reason why it is higher in genotype C, D, and A is because so the most of these genotypes were also overrepresented within the overall data set. But just to say that these mutations are present across all genotypes, really, and that has been because we included all data, and we included sequences across all genotypes. So we did not like specify on one or the other, but yes, it can be present across all these. But just to know that this data, the prevalence varies based on the number of sequences represented across all these genotypes.
Bundle of thanks.
Thank you, Jolynne. Any other questions from the audience? Which one? Post-lunch session? Very nice. Jolynne, I have a question for you. So it's you did a lot of systematic reviews and pooled data from a lot of different places. It's always difficult to do that, and you mentioned heterogeneity of data and everything. So besides that, what were the other biggest hurdles that you faced while pooling all of this data, both from the bioinformatics perspective as well as from the epidemiological perspective?
So the biggest challenge is really analyzing this data, and sometimes it's better to have like 2 or 3 people looking at this data set to ensure that it's accurate. Sometimes when you're dealing as an individual, you might get to miss maybe 1 or 2 things, but then so working as a team is quite crucial to like ensure that all the data is accurately gathered, or nothing is missing from that has been published. In terms of genomic data, it's also having like consistency. Again, just goes back again to the heterogeneity, consistency in how people define, and that's actually the biggest challenge in defining resistance. So with tenofovir, as we've seen, it's not just an individual mutation that causes resistance. You need multiple mutations coming together to actually say this is what causes resistance to this specific drug. But you'll get some studies only reporting one mutation, and that may not really be an accurate representation. So how do we, having a standardized method to report these mutations would be really ideal. And then also, testing. So there is one aspect, testing, and looking at the sequences and looking for these mutations, but also going to the lab to test whether, doing phenotypic testing to actually verify that is this actually a problem. Or sometimes you'd get like, yes, data has been obtained, analyzed, but the individual where the isolate or the virus was obtained from, the person was actually undergoing or responding to treatment very well, but these mutations can indeed be present. So that inconsistency in terms of like, how do you report for actual mutation is a huge challenge when it comes to reporting Hep drug resistance.
Thank you, Jolynne. So from the epidemiological perspective, did you identify, from some geographic regions where there was the lack of data or very little data, and how did you deal with, combining various kinds of populations, because this could have been done in some special populations, some general populations, etc.
Yeah, so for the initial review, we just pulled all the data together, regardless of where they came from. But we actually did look at to see which is the population that is actually contributing this data. So in this case, you get that most of them were HIV Hep B co-infected. So there is a huge gap across Hep B mono-infected individuals, and that is actually a major challenge because one, they tend not to get diagnosed, and they tend not to get treatment. And this is because the way the system has been developed within the African context, there's a lot of like mobilization and and focus on HIV clinics that are really well advanced. So if someone is HIV Hep B co-infected, they will be better treated. And so looking at the outcomes, you get that people who are HIV Hep B co-infected have a better clinical outcome compared to individuals who are HIV mono-infected, just because of how they access and interact with the healthcare. So in terms of the population, this is the key differing population. In other settings where you get like in North Africa or Southwest Africa, it's really again the aspect of like people being interested, or actually facilities and capacity to do hepatitis B work. You get that there are other priorities in some other settings where when you start talking about hepatitis B, they're like, oh, we don't have hepatitis B, but it's really because it's not actually been looked at in our population carefully to actually say to actually say whether it is there or it's not there. So you can say that a problem does not exist if you don't look for it. So that's one way of like saying there is no problem, but actually when you look, there is a problem. So there's there are those data gaps really just because of various priorities across these settings. But in terms of like a population difference, that is the key population diff. So HIV Hep B co-infected, and also really a key thing is so with Hep B hepatitis B, mother to child transmission is a key way of transmitting. So with Hep B, you get like when an adult, for example, is infected, they have a 90% chance of clearing that infection, even with immunologically. But when an infant, someone below the age of 5 years, is infected with Hep B, they tend to develop chronic disease, and then it tends to manifest as liver cancer at the age of an early adulthood. So looking at these differences of transmissions and how various populations are infected, that is the other something that is still being looked at, but quite exciting. Yeah.
Yeah, that's absolutely right, Jolene. So for many of these infectious, we tend to say that absence of evidence is not evidence of absence. So, once again, I would like to thank you very much on behalf of everyone in the room, and for a very insightful talk. So,
Thank you so much. Thank you so much for the invite.
thank you. Thank you very much, Jolene.